only last August. And yes, I forgot to mention that we are recording this meeting. So eventually we can put up the presentation on our website. So people who cannot join can always watch the presentation later. So yeah, our overarching goal is to bring together researchers who use ancient environmental DNA to advance knowledge of critical transitions in Earth's recent history. So more specifically, but not exclusively, we aim at establishing an international ancient eDNA network and apply paleogenetics to characterize past critical transitions. Jeez. Okay. So yes, if you want to learn more, I don't want to take up too much time, so you can always have a look at our short article that was published in the Pages magazine recently. You can also visit our website and register to the mailing list if you haven't done that already, so you can keep updated with our future activities. So we'll be planning workshops and other, so there's a seminar series. There will be other activities in the future, but so we're just getting started. And we're also happy if people want to get involved. Feel free to contact me or other members of the steering committee if you have ideas or if you want to participate in any way. You're very welcome to contact us. This is a group aimed at, getting people to work together and collaborate. So we're open to anyone who wants to participate. Also, if you are not a member of the Sedimentary Ancient DNA Society, you can also join. Visit the website. There's lots of information and many ways to get involved as well. So now I'd like to move on and present Charline Giguet-Covex. So she is a member of the PaleoEcoGen steering committee and also an advisory board member for the Sedimentary Ancient DNA Society. Charline did her PhD in sedimentology and geochemistry at the EDYTEM lab in France. Her research was focused on reconstructing past hydrological changes in the alpine area through the study of erosion dynamics. Charline did 2 postdocs, one at LECA in France and a second one at BioArch at University of York, where she gained experience in sedimentary ancient DNA to trace agropastoral activities and vegetation changes and to assess the impact of human activities on the landscape. So Charline is currently a CNRS researcher at EDYTEM in France and has a main interest in reconstructing past trajectories of mountain agroecosystems. So I will now stop sharing and invite Charline to share her screen.
Yes.
Yeah, can you co-host me so I can record it?
Excuse me?
Can you co-host me? So you click on the right part of my...
Yes.
Video, and you can mute Morgan also because, okay, she did it.
Yeah, so yeah, that's a good... I'll make Eric co-host, and yeah, just a last note, make sure you're muted through the presentation, and there will be a period for questions at the end. You can either write in the chat and we'll check the chat at the end, or just raise your hand at the end and I will try to manage the questions.
I'm not co-host yet.
You're not? Oh, sorry, I did it. More
maybe it's only one.
Okay, now it should work.
Yeah, perfect.
Okay, cool, thanks. Charline, take us away.
Yes, so do you see my screen?
Yes, we do.
Okay, thank you. So thank you for the invitation to start this seminar in the context of the PaleoEcoGen group, in the Pages group. So I wanted to to present a work about the spatial variability of past trajectories of alpine ecosystems. So it's a collaborative work that we started more than 10 years ago. So between my laboratory, EDYTEM, and also the LECA in Grenoble, and also the cartel in Chambéry and Thonon-les-Bains, and also in the collaboration with the University of York with the Department of Archaeology. So, so as you all know, the Alps has a long story of exploitation by humans due to the diversity of resources, which are related with vegetation zones, related with the altitudinal gradient. And people developed a vertical mobility and also adapted their land use related to these different ecological zones in the different altitudes, and also according to the season. As you know, mountains are very sensitive to climatic and more generally to environmental changes. So our question is, when and where did agropastoral activities develop, and what are the impacts and interaction with the environment? So to focus on this question, we will use the conceptual framework of trajectories and critical transition, because it's a topic, one of the topics of the PaleoEcoGen group. So we will try to discuss tipping points or resilience phases and resistance and adaptations. So we will focus in terms of condition on the human activity, and more specifically on agropastoral activity intensity. And we will study the response of the system, which is the vegetation composition and the erosion dynamic. So here I put some trends that we can observe in a transition or in shifts and in trajectories of an ecosystem state. So that is published by Schaeffer in 2009, and we will try to put in this context our results. So the method we used to reconstruct these ecosystem trajectories are based on sedimentological analysis and geochemistry for the reconstruction of the erosion. We also used mammal and plant DNA to reconstruct agropastoral activities and the vegetation dynamic, and especially we use the DNA because we would like to have more local records compared to pollen that are more regional. And we included some data from a coprophilous fungi analysis to have another record of pastoral activity intensity. And I will not show here, but we also integrated results from archaeological research to better constrain the history of human activities in the mountains. So here is a distribution of our study sites in the Alps, in the western part of the Alps, as you can see on the map here. So here you have the latitude. So we have a lot of study sites located in the northern part of the western part of the Alps, and some sites also in the southern part, but at relatively low elevation here. And here this dashed line in white here is representing the division between more internal massif compared to more external and subalpine massif here. So here is a synthesis to reconstruct the pastoral and livestock farming history. So based on the multiproxy approach to have a more robust reconstruction. So we can see a record of Plantago, that is an indirect proxy to reconstruct pastoral activity and intensity. We use also mammal DNA with the detection of cows that are represented in purple and sheep in pink. And for one lake, we also detected some goats in blue. And for the lowest altitude lake, the Lake Aiguebelette here, we detected some pigs that are in red. To complete this data set, so we also have for some lakes record of copophilous fungi that are represented in black. So it's one specific fungi, which is the Sporomiella. And for this study site, we don't have DNA data because of taphonomic problems, but we use pollen data for the Plantago, and we also have the Sporomiella record. So if we synthesize this data set, we can see that we have possibly first activities developed relatively early in a high altitude site during the Neolithic period. And so there are not very significant indices of these activities, so probably we have activities of low intensity during this period. At lower elevation, the Neolithic period show relatively well recorded activities with the presence of cows. And then in higher altitude, the first significant and robust detection of the presence of pastoralism are recorded during the mid and the late Bronze Age. So it's often difficult to detect low intensity activities. So for example, in the Neolithic, in the highest altitude lakes, with mammal DNA because of the low DNA production and the low DNA transfer, so probably related with taphonomic problems. During the Iron Age and the Roman period, we have an increase of the activities recorded in high altitude lakes, like this lake here in the lower part of the subalpine belt and in the upper part of the subalpine belt, and also in the alpine belt during the Roman period here. Then during the medieval period, we have a new development of intensive activities, so more specifically from the High Middle Age, so 1000 years ago, at the exception of the alpine belt, where we don't have development of activities. So during between the 13th and 15th centuries, depending on the study sites, we can see a change in animal composition with the development of cows compared to other animals like sheep. At the lower altitude site, we can see also the huge development of pigs here. So if we move at lower altitude, where we can also develop agricultural activities, so I will just present the results from the sites in the south part of the Alps, so which is called La Roche de Ram. So it's a work of Katarina Julias that is following the seminar today, I think. So here, so we have to take care about the HDEFs model that is not well constrained for the lowest part of the record here, but at least from the Roman period, the HDEFs model seems to be quite good. So here, during probably the Iron Age and the Roman period, we have first development of cereals here and an increase in cultivation of hemp. We also can detect later during the Roman period the presence of fruit trees like plum or cherries or peach with prunus here and apples. Then we have a new increase in the activities from 600 AD to 1650 AD, so from the early Middle Age to the beginning of the modern period, where we can see the huge development of fruit trees with, for example, walnuts here or grapes, and also the huge development of apple and plum or cherry or peach with the prunus again. We also probably have the development of pastures as reflected by this taxa here, but we are still waiting the data from the mammal DNA to confirm this trend. So during this period, we have an intensification from the late Middle Age here, where we can see the huge development of fruit trees with walnut and grapes, for example, and also the cultivation of cereals suggested by this taxa, but also by taxa associated with cultivated fields in blue here. We also can see the development of the crop of lavender. So then, for the last 4 centuries, we can see a decline of the activities. So based on this data, we try to synthesize the evolution of the activities against the altitudinal context. So we can see the altitude here on this axis, and here the different time periods. So the number of animals or of crops correspond to the intensity, the relative intensity of the activities. And when we have a question mark, for example, here, it's because we are relatively sure to have a pastoral activities development, but we don't know which animals are concerned by these developments. So what we can see is that we have an increase in altitude from the Neolithic to the Iron Age in the development of pastoral activities. We have also a development at all altitude during the Roman period here, so mostly for the altitude for the pastoralism, so during the Roman period. Then we probably have a decrease of activities in pastoral activities, especially in altitude during the early Middle Age, but the beginning of the development of cereals, especially at the lowest altitude site. The highest development that we can see at all altitude occurred during the High Middle Age, so from 1,000 years ago. And we can see the change in animal composition around the 13th and 15th century, depending on the site, with the development of cows, compared to the decrease in crops in the lowest altitude sites. For the south, here, during this period, which is not synthesized on this figure, but we have an opposite trend with a huge development of crops, and we still don't know for the livestock farming history. So based on this synthesis, we were able to develop like a relative scale of agropastoral activities intensities for each study site, and we used this relative scale to analyze the impact on plant communities. For doing that, we based on a non-metric multidimensional scaling analysis that we can see as an example for the Lake Anterne here. So then we determine the centroid for the different time periods, which is represented by the color code that you can see here. And then we draw like a trajectory following the time with a different centroid. And the size of the circle here, which represents the centroid, are corresponding to the intensity of the pastoral activities. So what we can see is that before the Iron Age, so during this part here, at the beginning of the trend, we can see that we have like a relatively linear relationship between the changes in the plant community, which are relatively progressive, and the increase in intensity of the activities. So we can say that we are in this type of relationship. And then during the Iron Age, so with the red dots here, we can see the first important shift in the plant community composition. We are totally changing in plant community composition. So we are more in this type of response in terms of transition. So we are changing the state of the system. Then again, during the Roman period, which is represented in black with a black dot here, we can see a new shift in the plant community composition, but with no no really change in intensity, or it's not very visible in our data, but probably we have a change in practices which could explain this change in plant composition. So here we are again more in this type of pattern. If we follow the trajectory toward today, so it's the blue dot here, we can see that even if we decrease the intensity of the activities, because we are in a context of agricultural decline now, we can see that we have no resilience in terms of plant composition, because we are still moving toward a new plant composition. So here maybe we are experiencing a critical transition during this modern period. So maybe this is due to a new driver which impact our community, for example, climatic change, or maybe it's due to a legacy effect of all the previous changes related to human activities. So if we look at the the different trajectories for all the study sites that are represented on this quite complex figure. So here you can see the altitude, so the lowest altitude sites are on the bottom part of the graph, and the highest altitude site is in the upper part, of course. So here we didn't represent the lake Egbelet because we have a very short time period recorded in this lake, so it's quite difficult to really analyze the trajectory of changes in plant composition. And you will see later the trajectories for the upper altitude site. So what is interesting to see is that we can see for all the study sites an important shift which occur at different periods depending on the altitude or the ecological zone. So on the upper site, the transition occurred relatively earlier, so during the Bronze Age or the Iron Age for the two study sites. And at lower elevation, it's more during the medieval period, so the High Middle Age, so quite late in this lower subalpine belt for the Lake Gers here. And in relatively late period also during the early Middle Age for the Lake La Tuile and also the Lake La Roche de Ram, which is more in southern position. So for the two lakes where we can have agricultural activities developed. So here we are in this type of pattern that I presented in more detail for Anterne before. So for Anterne is the only site where we can see no resilience in the modern period. So because all the sites are experiencing a decrease in agropastoral activities, except maybe the lake in the southern part here. But most of the lakes are showing a decline in agropastoral activities. But except Anterne, they are all going back to the previous condition, or start to go back to the previous condition, so where we can see like a resilience. So for now, we don't really know why Anterne doesn't really responding as the other study sites, but maybe it's related with the altitude and the fact that it's this lake is in a more external position and quite high altitude position compared, for example, to Vernet. So for the upper altitude site, so the Lake Savine, which is more in the alpine and nival belt, we don't see any trajectory, but so all dots for all colors seems to be everywhere on the graph. But we can see some exceptions with the yellow and gray dots, so the older dots which are all on this part of the graph, and the blue, light blue dots, which are on the opposite part of the graph. So these two different periods are corresponding to the extreme climatic condition of the Holocene. So for the blue dots, we are during the coldest phase of the Little Ice Age, and for the yellow and gray dots, they correspond to the Holocene climatic optimum. So on this side, even if we have an important development of pastoral activities during the Roman period with the black dot, we don't see a very huge change in plant composition. The main change are more related with important change in climatic conditions. Now, what about the erosion trajectory? So here I selected just some lakes because it's too much to show all the lake trajectory. But here we can see all the variability. So it seems that the trajectory are quite different from one side to another one, but we can see some similar patterns. So two different main patterns that I called classical pattern and unusual pattern. So for the classical pattern, it's more like a linear response of the system with an increase in intensity of agropastoral activity and an increase in the erosion response, or a decrease in agropastoral activity and a decrease in the response, so like resilience effect. We can see that on most of the sites with some loop, for example, here on Lake Savine or here on Lake Anterne, or this small loop for the Lake Gers here. And then we can see unusual pattern. So for example, at Anterne, we can see like what I call resistant pattern, which means that we have an increase, even a huge increase in agropastoral activity intensity, but no response in terms of erosion. So it is that the system is resistant to this force. On the two other sides, Gers and La Tuile, we can see what I called adaptation phases with an increase or the same agropastoral intensity, for example, for Gers here, but a huge decrease in terms of the response of the erosion. So here it's like if the system is adapting to this forcing. And this pattern, resistance pattern and adaptation patterns, are occurring during the Middle Age, so not exactly at the same time on the different sites, but all during the Middle Age. So maybe here there are some response of the society, of a pastoral, of a peasant society to this problem of soil erosion, because the soil are representing an important resources for them. So maybe they started to develop some strategy to limit the erosion. So for example, in agricultural system by building some terraces. For example, at La Tuile, we know that there is in the catchment all the terraces, but we are not able to date them. And maybe also this period for La Tuile, for example, correspond to the period where people started to develop fruit trees. So maybe these trees started to limit, to stabilize the soil and limit their erosion. And for the upper altitudes, so for pastoral activities, maybe people started to develop new practices which are better for the soil resources. So what is interesting also to see is that we have a very low resilience of the system on the altitude side, which means that today we decrease a lot the agropastoral activity because of the agricultural decline. But the system remain relatively low, relatively high in terms of erosion. So we can see that at Anterne, but I don't show here, but it's the same for Vernet, which is at around the same altitude. So here we have a low resilience compared to the lowest altitude sites, so for example, Gers and La Tuile, where we also decrease the activities, maybe less compared to the upper altitude sites, but we decrease the activity, and we can see a huge decrease in terms of erosion. So maybe these systems are a bit more resilient in terms of erosion compared to the upper altitude site. And this is maybe related with the reforestation dynamics that we can see in these areas. So in conclusion, what we can say is that lake sediments allow an integrative reconstruction of agroecosystem trajectories. So we can reconstruct the agricultural history and pastoral history, the erosion and the vegetation, and try to analyze them together. So lake sediment DNA brought new information by detecting taxa that are impossible to detect with other methods, so especially for the animals, and also for some crops which are difficult to detect with a more classical method like pollen. And because the lake sediment DNA give a signal at the local scale, so at the catchment scale, we are able to study really the interaction between the agropastoral system and the vegetation changes and the erosion changes. And this is particularly important in our context because we are looking at the different altitudinal zones. And in perspectives, so of course this model is just drawn by analyzing very few systems. So we would like to confirm this model by studying more lakes, and also more lakes in the southern part of the Alps to see if there is more variability if we compare the north part and the southern part of the Alps. So thank you for your attention. So if you have questions, I will be happy to answer.
Thank you so much, Charline. That was a really great presentation. Very interesting. I'm sure there will be many questions.
Yeah, and I'm sorry because some people already hear this presentation, so maybe not so much things new for them.
Well, there's a lot of information in there, so I'm sure everyone can be interested in some aspects.
I have a question for Charline. Laura.
Yeah, go ahead,
So,
Lara.
Yeah, just looking at your conclusion, you say that, and we know this is true, that lake sedimentary DNA gives a local signal. How local it is in terms really of meter from the shore for the vegetation, of course. I mean, because this is what I'm interested in. For the animals, probably it's difficult to say exactly. Yeah, how close to the shore they should be.
Yeah, for the vegetation and in our context, because it's a small lake and small catchment, which are very sensitive to erosion processes, I think in most of the sites we record all the catchment area. So all the places where we are connected by erosion processes and also by the water network. So it can
okay, so you expect even vegetation like 100 meters away to be represented because of this?
Yeah.
Equally well represented, I mean.
Yeah, more or less equally. But the main problem is that in system where we have very huge changes in terms of erosion, maybe you can increase, because usually we increase over the time the erosion dynamic, we can have an increase in terms of representativity of the taxa during these phases compared to previous phases. So we have to take care of that in the interpretations.
Yeah, thank you very much. Very interesting.
I think I will call the names I see three hands risen. I will call them in the order I can see them. I'm sorry if there's differences in the timing, but so Alex, after that Eric, and then Marco. So Alex.
Thanks, Charline, for an excellent presentation. Super cool data. Yeah, I was just wondering, I'm sorry if I just missed it, but did you talk about potential drivers for that big shift between the Neo and Bronze Age?
Yeah, maybe I was not very clear about that, but yes, the main driver for that is human activities, the development of pastoral activities in the catchment.
Oh, sorry, I just meant like, why did it change in altitude so much? They just like, why would they go up is sort of what I'm wondering.
Some question from me actually.
Why would, yeah.
It's climate? Do you think it's climate related, Charline, the fact that they went upper and upper in the height?
You mean why the change occur earlier in the upper sites compared to the lowest altitude sites? That's your question.
Why over time humans change their habits in terms of where they put their livestock and the cultivated plant? Do you think it's related to the climatic changes?
No,
it was warmer during the Roman period, for instance. It was warmer during the medieval one period too.
No, I don't think so. Because usually the development of human activities are the highest during the Little Ice Age, so it's not really related with the climate change. Maybe slightly during the Bronze Age because the climate is better, but it's difficult to say.
Maybe they were going away from people at lower altitudes, going up, and I don't know, just interested in speculating here. Thank you.
So Eric, did you have another question or did that answer your question? Sorry, you're muted, Eric.
Sorry, yes, I was saying it was a very interesting talk and I'm not sure I saw it before, so I learned a lot. My second question was about when you do the NMDS, did you include all the cultivated and non-cultivated plants to see the changes? Yeah, so it's kind of not biased, but you know what I mean.
Yeah.
You don't look at the real impact of human on the ecosystem. You look at how it transforms it and how that impacts the plant community. And I guess you will see the same pattern with the NMDS anyway. But it's interesting.
Yeah, that's interesting. I was thinking to try that also, to not really include the factor.
Thanks.
Okay, so Marco, you had a question? I see you wrote something in the chat, but maybe you want to...
Okay, no, that was a sort of separate question. Yeah,
okay.
Fantastic talk, really. So my question is with the coprolitic fungi, how certain is it that such fungi can no longer live in the sediment? And I mean, would be maybe nice to compare that with transcriptomes to see if they are still active, basically, or especially messenger RNA. Yeah, because we know now that ribosomal RNA can be quite well preserved. Yeah.
Yeah, that's a good question, an interesting question. Yes, these fungi are very well preserved, so it's quite robust record for the intensity of pastoral activity. And yes, it could be interesting to analyze the DNA of these species and the RNA to see if it's alive or not.
Yeah.
I see there was a question in the chat, so maybe we can answer that before Inga can ask the next question. So in the chat, we have Lucinda, who was saying, what a cool talk. Thank you so much. And she was wondering if you could talk a bit more about the proxies for erosion.
Yeah, I didn't really present that. Yes, the proxies for the erosion are, depending on the sites, they are mostly related with flood deposit where we record floods. So it's a flood frequency evolution and flood deposit thickness evolution. On other sites where we don't have any flood recorded, it's more related with the sedimentation rate, and the sedimentation rate related with silicated deposits. And also we use geochemistry, like for example titanium, which is a purely detrital element, not affected by weathering processes, for example.
Perfect. Okay. We still have lots of time for questions, so we'll go with Inga.
Thank you, Charline. It was great to see this talk and nice to see you again. I have a question about the quantification of the pastoral activity. Is that the sum of the different proxies you had, the Plantago, Spariella, different mammals, or how did you calculate that?
It's not really the sum, but it's a mix between the different proxies. So when I have the Copophilous fungi, I can use it really like an intensity record. But yeah, when we only have DNA from mammals and Plantago, it's more, yeah, it's less robust, I would say, because it can be also biased by some taphonomic processes. But yeah, I try to correct these processes. So it's just relative changes in intensity, and there are a lot of hypotheses behind, I would say.
Thank you.
Okay, so the next two questions, we have Ray Bradley and then Sarah Crump, and then we can go back to the chat where we have more questions. So Ray, please.
Yes, thank you. That was a very interesting talk. I'm not very familiar with the area, but my question is about the transfer of the signal from the landscape to the lake. Is that presumably there are streams and rivers that are flowing down slope into these lakes. So is there a signal being transferred from above into the lake from higher elevations, or are these closed basin lakes?
Yeah, it's a similar question as before. Yes, these contexts are highly connected system in terms of erosion. So you really, because the DNA we focused on, I didn't say that, it's on the extracellular DNA that is supposed to be fixed on detrital particles and especially clays. So if you have erosion, you really transfer the DNA from what you are looking for. So especially plants and animals. So yes, because most of these systems are highly connected in terms of erosion, with a high connection from everywhere in the catchment, you can have quite good record of the system.
Okay. And another quick question. If you're sampling at very high resolution, let's say, every few centimeters, how is the signal transferred from one sample to another? In other words, what is the persistence of the DNA record from down core?
You mean in terms of temporal?
Yeah.
You can have record a quite very long record with several millennia, and yeah. And
No, I mean, if you're sampling, maybe I'm not explaining this, but if you're sampling, let's say you're trying to get 100-year resolution in the sampling, will some of the DNA sort of hang around in the landscape and be transferred through the signal so that you're getting a kind of a smoothed record of what's happening in the landscape? See what I mean?
Yeah, you are speaking about a memory effect.
Yes, exactly.
This could be possible, I think, for more for trees, because I think the DNA from trees can stay quite long during a long time in the soils. So maybe for this record, you can have like a smooth trend with like a memory effect. But we didn't really discuss changes on this temporal resolution.
Okay.
Discuss because of that.
Okay, thank you very much. It was a great talk.
So Sarah?
Sarah?
Yeah, thanks. Thanks for the great talk. Really interesting. I have, some related questions building off of Ray's about taphonomy and catchment specific processes. So you mentioned, I think you mentioned early on that one lake had inadequate preservation. So I'm wondering, why do you think that lake had poorer preservation than the others? And then relatedly, if there are minor differences between the lakes in terms of sort of transport and taphonomy and each lake's sort of sensitivity threshold for recording, especially mammals, did you need to account for those differences in taphonomy between the lakes when you're comparing the intensity of agropastoral activity across different sites?
Yeah, it's difficult to really compare one lake with another one. It's more a comparison inside one lake and over the time. So it's difficult to say the intensity is much higher on this site compared to the other site, because the sensitivity of the site is different relative to several parameters like geology and erosion processes, vegetation cover, and topography and things like that. So it's difficult to compare one lake with another one. So I just try to have just a comparison from one side, but over time. And your question about the taphonomic problems that I mentioned on one lake. So it's one lake that is called Laoche. And this lake has nearly no catchment. So what we are able to record is plants that grow on the edges of the lake. So we don't have a very interesting signal in terms of plant composition and changes in this plant composition. So I already got some similar results on another lake with nearly no catchment around the lake and very flat catchment. And I thought it was because of that, but I was not sure because it was also a relatively acidic context, and I was thinking that maybe also this condition could be not very good for the DNA preservation. And finally, I tried again on another similar site in terms of catchment, topography, and size, but with another geological substrate, and which is no acidic. It's a calcareous environment, and we got slightly better record, but not very good record, only the edges.
Gotcha. Yeah, thank you so much. That's really interesting.
Yeah, that's why I really think that we are complementary with when we work with other proxies like pollens and copophilous fungi. Because pollens, for example, are not very good in very erosive system, because the pollen grains are slightly degraded by these processes. So it's good for DNA. And in other sites, it's not good for DNA, but very better for pollen. But not only local signal.
Great, thank you.
I see we have two questions in the chat. One is from Marco, who was wondering about the package you used for the NMDS combined with the driving forces.
It's the vegan packages on air.
I didn't get that. Can you repeat that? Or maybe put it in the chat. Yeah.
If I can repeat the response. I use R and the vegan package.
So I didn't know vegan could do that. Yeah.
Me neither.
I mean, NMDS, yes, but not the fancy other things.
I did it separately. Okay, I did the NMDS with the vegan package, and then I just draw the size of the circle for the intensity of agropastoral activities, just with Illustrator.
Oh, okay.
Maybe we can do that with R, but I didn't try.
Okay.
Tobias is wondering also if the Sporomyella, I can never say that word, Sporomyella fungi, are surveyed by morphology and not DNA in this study, right? The spores, you studied the spores just morphologically, not with the DNA.
Yeah, not with the DNA. You could try also with the DNA, but here it's just a morphological.
That rendered my question moot because I thought you did it based on DNA. Yeah.
Okay.
Maybe I can ask one last question. I don't see other hands. Last chance. I was just wondering, maybe you can just speculate on the. You mentioned that there was now this agricultural decline in the recent times, and I can't remember exactly, but there was at least some of the sites where there was like no resilience, and you mentioned that that could be because of climate, basically other drivers, or possibly climate, or also more like the accumulation of older drivers, and now there's a transition, even if the stressor seems small or inexistent. Can you say something about that? Like what's the role of climate in your
I don't know.
I mean, do you think it's a major aspect of, you know, the differences that you observe in the northern or southern latitudes, or also more alpine area versus the lower lands? Do you think that's...
I propose maybe the climatic explanation as a possible explanation, because the upper altitude sites where we can see impact of human activities. So I thought maybe there is something related with the climatic conditions, but I really think there is also a legacy effect, but I don't know how to study the role of these potential drivers.
Yeah. I agree. It's difficult. But yeah, so thank you, Charline. I think that we now have only 4 minutes left. That was a fantastic talk, very interesting. And for sure we could talk about it a lot more. We can announce already that the next webinar will be on February 24th. So same time, and I'm not sure if Trisha, are you there? Trisha, do you have the confirmation of the next speaker, or should we wait?
I do have confirmation of the next speaker. So it is going to be Zofia Teranu. And then following that, just waiting on the final confirmation, but we're hoping to have both March and April taken care of very shortly here. So keep an eye on your email and we will keep you up to date. But Zofia has agreed to be our next speaker for that.
Perfect. Yes, so for anyone who's not yet subscribed to our mailing list, so we will announce the seminars via this sedaDNA Society always, but also the PaleoEcoGen mailing list. So if you haven't subscribed yet, you can always do that. Just visit our website. There's a link. I promise not to send too many emails. What else? Eric, Trisha, more announcements? I think that's all of it for now.
Yeah, I think that's good.
Okay.
Perfect.
Great. So thanks again to Charline.
Yes, thank you for all the questions. Very interesting questions, and I hope to discuss more about critical transition soon.
Yes.
So what is the journal you submitted? REC?
Regional environmental changes.
Okay, thanks.
Yeah, not all the sites. Not the site in the south and not the lowest altitude site in the north, because of the time period that is too short.
Yeah, I'm sure it deserves better. So I guess you try some journal before.
Sorry?
I guess you try some other journal.
No, I didn't.
You went there, okay.
Yeah, because it's a special issue
Okay.
related to a project. So yeah, I said okay.
Yeah, so maybe we can stop the recording if people want to stay for another few minutes just to chat.